Today we will discuss about restriction enzyme. So restriction enzyme are basically those enzymes which can cut the DNA molecule into a small fragments. Now, restriction enzymes are also known as molecular scissor because they do act like a scissor. They cut the DNA into different type of fragments. So some basic terminology used in the topic of restriction enzyme. Uh, these are palindromic sequence recognition site, sticky ends, and blunt ends. But they are very simple terminology. Now, I want to explain the palindromic sequence that what are a palindromic sequence. So, palindromic sequence are those type of sequence which we can read from either from upstream or from downstream. So, the EcoR1 is basically bacteria from which the restriction enzyme are obtained. So they cut on this type of sequence. This is the cutting sequence of uh, Escherichia coli enzyme. Now, if you see this example, so uh, we we read this sequence either from upstream or either from downstream. So if we try that GAATTC and GAATTC, they are become similar from either upstream or from downstream. So this is the palindromic sequence. Now the second terminology which are used in restriction enzyme topic is recognition site. So recognition site are those site which the restriction enzyme are attached and they do cut on the recognition site. Suppose this is the example of EcoR1 restriction enzyme which came from the Escherichia coli strain of bacteria. So this EcoR1 restriction enzyme are come here and they do cut on this G and A center of this nucleotide. So they make a different type of ends like sticky ends, blunt ends, or cohesive ends. So this is the recognition site in which the restriction enzyme are attached. So we we call them as a restriction enzyme, recognition site. Sorry. Now the third terminology are used in the restriction enzyme topic is a sticky end. So the example of this is also example of sticky ends. Now, if this is the a sequence when restriction enzyme are came here in this recognition position, so they make cut here. So one is this cohesive in or palm, and another are this type of cohesive in or palm or sticky in or palm, which we use another type of enzyme ligase, so we can combine this type of sequence with each other. So this is a sticky in. Basically, this is a structure of sticky ends. So Here, two types of sticky in are found. So these eames are called sticky eames are cohesive eames. Now, the another type of enzyme that is basically SMA1, which can produce blunt end. Now, let's see to this palindromic sequence. When the SMA1 enzyme are uh, attached to this recognition site, so they directly cut on this central point and make two blunt ends like this. So here they do cut on the center. So two types of uh, ends are prepared from the SMA1 restriction enzyme. So each and every restriction enzyme have different type of recognition site and restriction cutting site. So this is the recognition site of EQR1 enzyme, and they make cohesive in our um, sticky in, and the blunt ends are preferred due to the SM1 restriction enzyme. Now let's go to the other um, subtopic of this restriction enzyme, which is basically known as nomenclature of restriction enzyme. So how we can do naming on restriction enzyme. So this is a restriction enzyme bacteria is equal R2. So the E represent the genus and CO present the species name. So here the E are Escherichia, which are basically the genus name, and Co are Escherichia coli. Coli are basically the species of that bacteria. R stand for strain, and to show that the discovery number that they are discovered second time. Now the let's move toward the application of a restriction enzyme that where restriction enzyme are used. So in molecular biology there are some basic test and some basic technique which the restriction enzyme are do work. Now the first one is DNA fingerprinting. So we use restriction enzyme and the process of DNA fingerprinting. The second use of restriction enzyme are in southern blotting, which we can identify the DNA molecule, which can we do for the DNA deoxyribonucleic acid. Third one is RFLPs, restriction fragment length polymorphism. So in this method also the restriction enzyme are used. Now the other example of restriction enzyme, they are also used in some PCR techniques. So this is all about the restriction enzyme, which we can explain the palindromic sequence recognition side, sticky ins, and also the nomenclature of a restriction enzyme, that how we can uh, give the naming to the num to the restriction enzyme, and then we shortly explain the application of restriction enzyme. Hopefully you understand this lecture. Thank you. See you next time.